So, welcome to all attendees. We are going to start this side event on microbiome at the heart of One Health, from soil to plate and beyond. And if I understood well from the summit, that the World Microbiome was not pronounced so much. So we are going to, through this event, to explain a bit more what is microbiome, using different angles from policy point of view, regulatory, scientific, of course, so that everybody has a clear vision of what it means and why it's important in the One Health concept. I thank the people who came here in person, and I know that in line we have lots of people who register, more on about 160, so I welcome them also. And we will start the event because we were a bit ambitious, so we have lots of speakers and tight timing. Bonjour. So I'm honored to welcome to start this meeting our CIO, Philippe Maugin, who will introduce this event through a short presentation. Philippe, the floor is yours.
Thank you, Emmanuelle. Good morning, everybody. I have some sound bizarre, but maybe it's good for you.
Fine here.
It's okay for you. Okay. So, dear colleagues, dear attendees, both in Lyon and online, I am delighted to open this side event, organized by INRAE and EFSA, and also with the support of INSERM. We share a common vision on One Health and the role of microbiome science with EFSA and INRAE, and also with INSERM. And as you may know, we coordinate the World Microbiome Partnership initiative. I don't have too many times to detail, but I just want to stress on the fact that for INRAE, the study of the microbiome is a long-standing activity, starting from so many years for animals, and particularly cattle, but also for human and for soil. And we were supported by 2 Horizon Europe projects, and this work has led to important scientific progress, publication, and a number of innovations. Indeed, research on microbiomes is still rather young, but already numerous scientific publications, international organizations confirm that the transversal nature of microbiomes strongly aligns with the One Health approach, serving as a biological link between different ecosystems. And that's why we are organizing that side event within the One Health Summit umbrella. The microbiome is a trigger, but also a solution for major society challenges, like the increase of chronic disease, which are the leading cause of death worldwide since 2019, but also the development of antimicrobial resistance, biodiversity loss, climate change, and the transition of agriculture and food system. Alongside other academic partners such as INSERM, INRAE is committed to translating scientific advances into public policies and societal changes. That's why we launched several international research initiatives over the past 5 years. One of them, the World Microbiome Partnership, led and organized by our colleagues, Joël Doré and Emmanuelle Maguin, was initiated in 2023 with the idea to explore at the world level the diversity of microbiomes. In June 2025, under the high patronage of the President of the French Republic, we launched the World Microbiome Partnership officially. We had also a scientific international agenda. We set up working groups, and we signed with a lot of partners the Paris Declaration. And this collective commitment to advancing microbiome science is, for me, a strong lever for innovation, for policy, and also for global development. We now gather more than 200 members, and we have already achieved the first goal of bringing together professionals, as we do today with that scientific meeting in Lyon, to facilitate innovative partnership and to promote harmonized global regulatory process. As part of the One Health Summit, as you may know, more than 300 scientists gathered in the One Health, One Science Symposium this Monday, made several recommendations for One Health approach, and one of them was to establish a global interdisciplinary microbiome observatory with the objective of positioning microbiomes as a strategic lever for sustainable development across all sectors, agriculture, livestock, food processing. And that's why, in response to this need, to this call, we are taking together action by creating the Global Observatory for Microbiome Health, with the objective, the goal to collect 1 million human gut samples by 2030. I'm really proud and happy to launch with you this morning, concretely, that new World Observatory. The initiative began with the establishment of the Alliance for Human Microbiome Cohort in 2025, coordinated by the World Microbiome Partnership. As of today, the Human Microbiome Cohort Alliance has already gathered 500,000 samples, half of the objective, from coming from more than 50 partners in more than 25 countries on the 5 continents. So it's really exciting to have such a good development today. And so that's why our objective to collect 1 million gut by 2030 is already well engaged, and I'm really confident in our future success. You know that human microbiome is now recognized as a major determinant for health, intervening in several diseases like inflammatory, cardiovascular, neuropsychiatric diseases, or some cancers. The change in dietary habits, the rapid urbanization, the loss of biodiversity also make microbiomes a strategic indicator of the impact of this transformation on human health. That's why our observatory for microbiome health is a strategic new infrastructure at the world level, which will firstly map, harmonize, and analyze data on human microbiome. We hope that from 2028, it will start to reach for microbiomes from food, animals, plants, soils, and oceans. This will allow analyzing the network of interaction between microbiomes and its impact on health from ecosystem to humans. Among other research topics, the observatory will be key in measuring the impact of the environment and food system on human health. Secondly, to creating a common international scientific standard. And then third key target, transforming innovation in the field of microbiome to reshape food system and develop new therapies. As you may know, the markets for microbiome-based products in human health is expected to be worth $1.4 billion in 2027. And the global market for agricultural application will be maybe worth $12 billion, including biocontrol products, microbial additive for animal husbandry, restoration of soil, etc. I'm glad you have gathered here today to discuss how microbiomes can help address the challenges of antimicrobial resistance, crop productivity, animal health, food quality, nutrition, and human well-being. I'm convinced that studying the food system as a whole will reveal how improving microbiome health in interconnected ecosystems can contribute to food security, to reduce environmental pressures, and to promote long-term planetary health. Thank you for your engagement towards this observatory on the scientific side, but also on the policy side. This ambitious international project needs the engagement at all scales from governmental and funding institutions willing to support its development with the aim of fostering the dialogue between science and policies. I wish you all a fruitful event and excellent exchanges both during and beyond this event. Thank you very much.
Thank you, Philippe, for this insightful introduction, grounding in the past and opening perspective for the future. And it's now my pleasure to introduce Carlos das Neves. I'm not sure about the pronunciation. Okay, thank you. Who is Chief Scientist at EFSA and member of the One Health high-level expert panel. And we are very pleased and delighted to host him. And here is the something.
Merci, Emmanuelle. So, cher Philippe, Emmanuelle, dear friends and colleagues, I truly hope you've been enjoying. I hope most of you have been enjoying the last 2 days. As this summit is coming to an end, I have to confess, this morning I was thinking, the last 2 days, a lot of things was happening. We were all sitting in these auditoriums, and you keep looking at the phone, and you see, civilizations about to be wiped out, and everything that is happening. And it came to mind to me a sentence from Albert Camus. This au milieu du l'hiver, j'ai découvert en moi un invincible été. Which is in the middle of all darkness, I found a unique resilience to keep moving forwards. And I think we achieved that yesterday. And I would even challenge that I think we found space for microbiome there. Not to mention that, as our president of INRAE has just mentioned, President Macron made it very clear that the observatory is a go. So I think we found the spot for it in our narrative. But like any good narrative, once we want to move from words to action, and this summit was all about going from words to action, we have a few challenges. And I don't need to praise the importance of the microbiome. It's already been done here, and its transversal impact and importance for all living elements, all the sectors of our One Health village. So you allow me to be a bit l'hiver and focus on the dark sides, or on the things that we really need to keep up going. I want first just to congratulate the organizers and say that for us, EFSA, this is important. It's important also because we want to make words into action, and we signed a beautiful and very informal memorandum of collaboration with INRAE just last year. And it said we're going to do something on a few topics, microbiome one of them, and we are here to fulfill that promise. And my colleague Constantino's here in the front. He runs the microbiome, he carries the microbiome flag for us. And we will meet again even later this year when we have another organized EFSA INRA workshop dedicated to regulatory aspects of microbiome. I've heard it's in Versailles, to be checked, so that sounds very royal to me. It's good. A date to be set. I need a date soon so I can start advertising it. But before here and there, also to tell you that this is important for EFSA, it's part of our strategy, and it's important for the Union, who has made a strong commitment in its life science strategy, as some of you are aware, of having microbiome as one of the flag elements of the new European life science strategies. Horizon Europe is pledging another 100 million euros for the upcoming and last part of the program. It's been put into the strategic priorities of FP10. Many of you are doing front research. I see EMBL here, advancing the field as quickly as you can. Now let me give you the bad news. And all these companies that are wanting to using the microbiome to do the food products and the feed products and the medicine products Once they send their beautiful dossiers for approval and safety checks, we're going to tell them that most probably, should never be totally certain, but we most probably going to tell them we have no idea how to evaluate this. And this is because, ladies and gentlemen, the gap between scientific knowledge on microbiome and our capacity to translate it to regulatory use, this gap is big. Perhaps not as big in gut and other aspects of microbiome as it is in environment, but you remember that we also need to look into the microbiome every time we evaluate pesticides. But there is a gap. And my plea is that through these collaborations, through the World Partnership, through the Observatory, we start trying to close this gap. We have a few ideas. I leave that to Costas, so I don't start saying stuff I don't understand about. But my plea is the gap needs to be closed. We are in a push for stronger competitiveness, stronger innovation in our European Union. We want people to make transitions to sustainable and better lifestyles. And I think you all believe and lobby, microbiome is part of that change, of that transformation, then please help me, regulator, make it happen. Otherwise, we will stay in the winter of Albert Camus and we will not see the light. But there is a lot of light here in the room and online, so without further delay, thank you to the organizers, thanks to all of you, and good luck forward. Merci beaucoup.
Thank you very much for this introduction and for the light coming. I think, when I was young, there is this song about, sun and all the stuff, so remind me something. And now it's my pleasure to welcome Eric Cardinale, who is a colleague from ANSES. He's the scientific director for animal health and welfare, and he's a coordinator of the interagency One Health working group, and he was really instrumental in the organization and preparation of this meeting. Eric, the floor is yours.
Je vais rester debout, merci. So good morning, everyone. I'm really happy to be with you today and to open this session with Philippe and with Carlos. And I will talk just a little bit about microbiome. And of course, I mean, this summit was organized by many very nice people. You see head of states, head of international and regional organization, and so on. But final goal is to accelerate the implementation of this One Health approach. And very nice objectives as well. I mean, like international and interdisciplinary dialogue, reshape global institutional framework, and so on. But what about microbiome? And what is useful to work on microbiome, and why within this One Health approach? Well, during this summit, and especially, I mean, during the first day for the scientific conference, we addressed 4 thematic areas. So, I mean, emerging infectious diseases, AMR, sustainable food systems, and pollutions. And we realized, looking at those thematic areas, that microbial communities are really crucial for many facets of health and resilience, but also from human, but not only from human, but also for plant, animal health, to disease resistance, ecosystem adaptability, planetary climate regulation. And so we see that we do have a microbial continuity. Microbiomes, as you know, are not isolated at all. I mean, there is a constant state of flux and exchange. And One Health, when we look at microbiomes through the One Health perspective, we see that bacteria and soil can influence directly the gut health of a human or an animal. And so we have a link between the soil and the plant to the animal and humans, and of course, from the environment to all as well. And looking at that picture, I mean, I like this because, I mean, we see globally the microbial life spiral between the 3 compartments, environmental, animal, and human health, and we can develop and implement key One Health actions like, and it's only examples, of course, sustainable agriculture, genomic surveillance, bio-integrated urbanism, or cultivate microbial diversity. And so what could be finally the added value of working on the microbiome? So of course, I mean, we have this shift, and we try to focus on this particularly. I mean, the shift from reactive medicine to proactive ecosystem management. And like this one, I mean, the first one would be mainly on combating antimicrobial resistance, but also, I mean, improving food security and nutrient density. And of course, zoonotic disease prediction. And as you know, I mean, many EID like COVID-19, avian flu, or Nipah virus originate in animals. And you know, when you try to monitor this microbial spillover between animals and humans, I mean, you need to work at the interface between wildlife, domestic animals, humans, and finally, thanks to this microbiome approach, you can identify potential pathogens before they jump the species. Well, so, I mean, only some example, but it seems to be very soundful. I mean, how can we use this knowledge for a sustainable future? I mean, as an example, we can have the regenerative agriculture. The idea is to finally restore soil microbiome health to produce more nutritious food. Bio-integrated urbanism. I mean, the idea is to create urban green spaces to enrich, finally, microbial biodiversity and to strengthen, at the end, the immune systems of city dwellers. And of course, as I already mentioned, genomic surveillance and using DNA sequencing to monitor this emergence of zoonotic diseases. And finally, maybe we could move towards an all-audience perspective. And through this One Health approach, we can view humans, animals, plants, not as individuals, but I mean the host organism plus their associated microorganism. So at the different level, of course, soils, plants, animals, or humans. So what can be done as priorities? Of course, from the World Microbiome Partnership, we see that we do have a lot of strategic priorities, but also technical and scientific priorities, like standardization, regulatory harmonization, data stewardship, of course, and of course, environmental monitoring. And Philippe talked about it, but I mean, you know, set up microbiome observatories. For this and to address this particular topic, France has different tools, and I can mention two of them. And the first one is the France 2030 investment plan. I mean, and particularly, we do have a program called Food Systems, Microbiomes, and Health. And the goal of this program is how to translate the international One Health framework into concrete and scalable action. And through this One Health vision, what we try to do is to struggle with siloing. I mean, we try to connect, not to break, Carlos, right? To connect the silo and not to break the silo. And we try to emphasize this microbiome link. And of course, at the same time, we keep this preventive sovereignty. And thanks to this France 2030 investment plan, we try to work on key pillars of implementation, like the scientific excellence, particularly with the integration of environmental data with clinical outcomes, on industrial innovation, and particularly the goal is to reduce chemical inputs in the food chain to protect biodiversity, and of course on public policy as well. The goal is to ensure healthy food and accessible and sustainably produced. And another tool, I mean, that's the one I'm trying to coordinate. I mean, this interagency One Health working group. I mean, we created in France the program agency. I mean, these program agencies are really the conductors for French research nationwide. With a prospective goal. I mean, the goal is indeed to forecast and prepare, to be prepared to scientific challenges. This coordination, again, connecting the silos, and directed research. And we have 3 agencies so far that are included in the working group. The one on health, supported by INSERM, the other one on biodiversity, supported by CNRS, and the last one on animal health and livestock sustainability, supported by INRAE. And on this group, we have 40 institutions, more than 150 researchers, and we try in this group to promote One Health approach and show the added value. like, on microbiome, as an example. And finally, at the end, structure, the French One Health research. In this context, we try to make the microbiome research not staying in a lab, but trying to bridge the gaps between the soil, the gut, and the society. And as a conclusion, well, we have to protect the microbiome of the soils because, we have a direct consequence on animals and human health, and we have a call to action. The idea is we must shift from a mindset only focused on eradicating microbes to a one focused much more on cultivating microbial balance. Finally, health is one, and it begins with the infinitely small. Thank you.
Thank you, Eric. Is there a question for Eric?
For later.
Okay, later. Thank you very much for this presentation. You set the scene perfectly. Yeah. And so now we will we are honored to welcome a recorded contribution from Junxia Song, who couldn't join today, but sent us a message and a recorded contribution. And if there is any question, she mentioned that she will be pleased to answer by mail to any questions.
Dear experts, friends, ladies and gentlemen, it's a great pleasure to speak with you today. Apologies, I'm not able to join you in person for this very interesting topic, especially just after One Health Summit, where global leaders, scientists, youth, and different stakeholders reaffirm their commitment to move forward One Health agenda, and also focus on so many excited deliverables. It's a perfect timing to talk about empowering microbiomes for the benefit of One Health. Today, I will briefly link microbiomes to agri-food systems, highlight the key gaps we are facing, and then share practical some entry points and a way forward for policy and action. Let me start with a bigger picture. Feeding people today is not just about producing more calories. It is about healthy, safe, and sustainable diet. We are already feeding billions of people, and the demand for food will keep increasing. So at the same time, unhealthy and unsafe diet and malnutrition are major drivers of disease and premature disease deaths. This is where One Health really matters. Our food system link human, animal, plant health. Microbiomes sit right at the center of this link, shaping how food is produced, how safe it is, and how it affects our health. In agri-food systems, microbiomes matter everywhere. On the screen, you can see animals, plants, soil, water, food, and even the air around us. They shape health, productivity, resilience, and environmental sustainability across the whole agri-food system. As we said, we can see the linkages, the central role of microbiomes connect different health domains in agri-food system. So microbiomes can be a very practical lever for One Health. When they are healthy, we can reduce the need for chemicals, for antimicrobials, and build more resilient agri-food systems. Microbiome-based innovations can help mitigate the risk of health risk and environmental risk, such as probiotics, biofertilizer, and biopesticides. Microbiomes in agri-food system and One Health have the great potential to advance many of the SDGs. By integrating microbiomes into agri-food system and One Health strategies can help improve health, supporting sustainable farming, boosted by economy, build climate resilience, and protect the environment. How microbiomes can be turned into practical One Health action. There are few entry points. Microbiome data can strengthen surveillance by spotting early signals of disease, AMR, and environmental stress. Microbiome-based solutions, as we just mentioned, such as the probiotics, by pesticides, it can help reduce the risks of the health and also reducing reliance on antimicrobials and chemicals. So third, we can improve sustainable practice for preserving and restoring microbiomes. So microbiomes data can also allow more targeted intervention. So FAO has been supporting this work through multidisciplinary collaboration and global partnership since 2019. Those activities are including conducting comprehensive literature review, publication, contributing to the Microbiome Learning Network, and which bring FAO together with 120 partners, and also ensuring the participation in a global conference. Here, just highlight that microbiomes are already relevant across major FAO priorities. You can see the links to nutrition, food safety, and bioeconomy, and also strong relevance for livestock and sustainable ecosystems. So the point is that microbiomes are not a standalone topic. This is cross-cutting work that strengthens what FAO is doing across programs and products. So this is the gap we need to confront. The One Health approach often remains pathogen-centric, focused on specific organisms that cause disease or on the burden to public health or production. At the same time, microbiomes remain largely absent from One Health policy framework. There are some key One Health challenges involving microbiomes, such as awareness of microbiomes are still low among policymakers and public, and governance is fragmented across human, animal, plant, and environmental sectors. We also lack standard tools and indicators, which makes it hard to turn science into action. In many low and middle income countries, technical capacity and infrastructure remain limited. On top of that, regulatory uncertainty, competing priorities, and limited resources mean microbiomes are often overlooked. The good news is that we have a real window of opportunity. After COVID, there is strong momentum towards prevention, growing urgency on AMR, and more focus on sustainable food systems. We already have a platform One Health connects sectors aligned with SDGs, so we don't need new structure. The science is advancing fast, and new technologies also allowed us to manage microbiomes at scale. So, in short, the conditions are ready. What we need now is policy uptake and coordinated action. So here's the practical way forward. First, we need to bring microbiomes into existing One Health framework. So health is managed across sectors, not in silos. Second, we need to stronger international collaboration to improve governance and share knowledge. Third, we must invest in research, education, and training to turn evidence into real action. Fourth, standardized monitoring systems are essential to track microbiome changes and give early warning on AMR, disease emergency, and ecosystem stress. And then we need clear stewardship and regulatory frameworks so microbiome-based solutions can be used safely and at scale, especially with strong capacity building in low and middle income countries. To close, my key message today is very simple. If we want to truly operationalize One Health, we must see microbiomes as part of the system, not as just a side topic. If we keep them invisible in policy, we will continue to react to problems rather than prevent them. But if we integrate microbiomes into One Health strategies, we can strengthen prevention, improve efficiency, and deliver measurable gains across ecosystem. Thank you very much for your attention.
So thank you for the contribution, and thank you for all the contribution that we had already, because what I appreciate is that you identified clear gaps and way to go forward, which is exactly what we need in this kind of dialogue, to agree on what we need to work on. So now, I don't know if he's online. We are going to welcome Pierre Mathieu Pelissier, who is a project officer at the Directorate General Research and Innovation at the European Commission. And he will, I guess, open some of the avenues for the future, talking about microbiome in European One Health policies. I don't know if he's... Is he online?
I'm online.
Yeah.
Okay, all right. The video is
So
delayed.
the micro is yours, Pierre Mathieu.
Perfect. All right. Good morning, everyone. Pierre Mathieu Pelissier, indeed, working at the Directorate of Research and Innovation at the European Commission in the unit on bioeconomy and food systems. And to complement the presentation that were done earlier this morning, I will mention microbiome in the European One Health policies. So, One Health has represented a strong paradigm shift in the policies that we have here at the European Commission, not only at the microbiome, but at a more systemic level as well. And it became a requirement across all EU agencies. It focused on human centrics, but now it shifts toward ecological one and a more systemic approach. So how does it apply? Well, one of the main things that we did is that we organized a task force, a new cross-agency One Health task force that unites the European Environment Agency, the European Chemical Agency, disease agency, medical agency, food agencies, and so forth. And the idea is that it represents the fact that those different agencies cannot work in silos anymore into their respective domains of application. The European Food Safety has to work with the European Environmental Agency and European Chemical Agency to develop the risk assessment protocols and so forth. And the idea is to integrate data collection and to create strategic joint scientific advice. So we are breaking the silo of One Health at the European Commissions using the One Health framework. When it comes to microbiome, well, last year in July, we published the European Life Science Strategy, which emphasized that microbiome were at the heart of the One Health approach. It's mentioned per se in the Life Science Strategy itself. It comes together with an announced 100 million euro in funding available through Horizon Europe for One Health microbiome solution. And an important aspect is that it's not only human health microbiome, but it covers a lot of other core priorities that drives industrial sustainability, climate resilience, and the restoration of degraded habitats, notably through food systems. So it resonates a lot with the presentation that were before me, where indeed One Health microbiome is applied to ecosystem, to different domain, and not only One Health or gut microbiome, but to environment, bioremediation, agri-food, and so forth. So this was announced in the life science strategy. It's reflected in a 100 million funding, and if you read more in detail the topic that cover microbiome and with the One Health framework in the work program that we have, whether it's the EIC or Cluster 6 of Horizon Europe, We have topics that cover food system and agriculture with holistic research innovation and commercializing new solutions that are based on microbiome. We also fund AI-powered foodome characterization to map components in foods, including microbiome, soil and livestock health, tracking AMR in soil to boost livestock sustainability. I will come back a bit on that later. Environment industry with a lot of bioremediation and aquatic pollution, leveraging biotech and microbiome solutions. And early cancer prediction, but links to food system and developing data repository that are fit for One Health application. So we emphasize on microbiome in our policies, whether it's on funding, but also it's also embedded into different policies, as you will see that we created. And it's reflected, for instance, in the soil monitoring law. It's a directive from last year, which is the first one to treat the soil as a living organism and ecosystem rather than just a substrate for crops. What does it mean? Is that it mandates monitoring the soil health, not only on a chemical basis, but also looking at biological metrics, the living organism and the living biomass that is in soil. There are technical working groups that are developing a protocol for that. And early 2026, for instance, one protocol using DNA metabarcoding has been developed to monitor microorganism life and fungal life in the soil under this law. We also have regulation linked to carbon farming, for instance, the carbon removal certification framework rules were expanded and were revised to better reflect the fact that microorganism and living biomass is also critical for carbon sequestration in soil. And in the EU Biotech Act, you may have noticed we are working towards fast-tracking microbial biostimulants, microbial biopesticides, who supports plant health, supports soil health, and indirectly human health as well, because those solutions have less residues and less impact per se on the food value chains. So it's funding, but also it's supported by the different policies that we have. It's also the case for animals and microbiome. So the new policy cycle marks the end of the blanket antibiotic use and the start of precision microbiome management. So how is it reflected in incentive and in guidance, where, for instance, the use of prebiotics or insect protein to prime neonatal microbiome in livestock and increase their natural immunity, hence reducing clinical intervention, is here to support this One Health microbiome in the animal side. As well as metagenomic surveillance to map the resistome. So we are tracking the AMR and the microbiome in the animals to protect the human food chain, because everything being linked, the human microbiome is affected by the animals' microbiome as well. And the environment and microbiome policies are also supporting this microbiome approach with urban early warning systems. So we have the wastewater directive, and its revised wastewater directive requests large city with 100,000 inhabitants more to monitor antimicrobial resistance gene in wastewater. And so it uses basically the wastewater system of a city as a health public radar overall. We also support industrial biomanufacturing and circular economy with policies again, so funding and policies. Regulatory sandboxes, for instance, where we can test synthetic microbe to degrade PFAS, or to produce biofertilizer that prevent river dysbiosis, which is the imbalance of microbiome composition of river, right? An overreaching of a certain kind of microbiome over some others. So as I presented very briefly and very synthetically, we support microbiome for One Health and non-food application via policies, but also funding, so two parallel track. And this is a high priority now for the European Commission. So as we are working towards the development of those initiatives, translating microbiome health, especially through the One Health scope, require a lot of metrics, aquatic indicator, KPI, but also capacity to connect the ecosystem. And the initiative that presents today, for instance, one of these initiatives that is really supporting this. We also fund interactive interoperable biobanks, and it's a systemic effort, and we need scientific data to build the next generation of One Health policies, and those initiatives are helping us a lot in these actions. So I'll stop sharing. Hope it was all clear.
Thank you, Pierre Mathieu, for this talk and for describing this European framework on what you're doing already and what you are supporting for the currently and for the future. Is there one question for Pierre Mathieu from the room? No? Okay. So thanks a lot again for this contribution, and we will go to the next speaker from EFSA. So our next speaker is Konstantinos Paraskevopoulos.
Very good.
Thank you. I trained a lot. I still got He's a scientific project coordinator at EFSA, and will tell us about microbiome.
Thank you very much. Okay. So, yeah, Carlos alluded to what I'm going to say. I will give you the details of how we see microbiomes at EFSA, and give you some information on basically the picture of what a food safety authority would care for when it comes to microbiomes. So, bear with me for the next few minutes. Okay, so first slide to tell you why it matters for us and where we see the relevance. Here, you can see, is there a laser? No, there's no laser here, no? Okay, yeah, so here this helix represents the remit of EFSA, yeah? So all the scientific domains that we deal with. As you can see, it varies from looking at products such as food and feed additives or novel foods or GMOs, all the way to plant health, animal health, environmental, and of course, humans. So that by itself shows you that EFSA is in fact quite a One Health organization already. And here on the side of this helix, you can see where microbiomes are relevant. It's pretty obvious that they are connected to most of what we do. So putting those two together, I think it maps quite nicely that microbiomes and the work of EFSA actually are well within the One Health spectrum. And microbiomes are really an excellent biological connector. So really, as I said, a perfect match, I would say. Okay. What is the issue, however, with regards to what's happening, what's being developed, and how we can deal with it as a food safety authority? So focusing here on the products, we see a lot of different products. They were mentioned and nicely presented by the previous speakers who have quite a variety, who have advanced feed additives, such as competitive exclusion products. We have next generation probiotics, we have environmentally and agri-food related products such as biocontrol agents, biostimulants, and so on. So it really is quite an active and fast-growing pipeline of product innovation. Putting this against what we can do today,
we have quite a few, let's say, challenges. So first of all, the current systems that we have were built for single strains. Yes, so they're not really fit for purpose to deal with complex microbial communities such as microbiomes. That itself, it presents additional challenges more on the technical level. For example, we don't really have the methods to be able to understand and assess the interactions. Yes, you have one community that is a product which will interact with the host community. What happens? Yes, we don't really have the methods and the data to be able to assess that. Very importantly, we are not able to connect from the exposure. We are not able to understand, to be able to tell when a microbiome disruption, perturbation, leads to adverse health outcomes. Yes, so not every change is a concern. So we really need to pinpoint when a change is severe enough, or let's say relevant, that it will become an endpoint for a safety authority like EFSA. That was explicitly highlighted in the recently published guideline for microbiological products. That was, I think it went out a few months ago. I think it was in autumn of last year, where it was made really explicit that we're lacking those standardized assessment frameworks to be able to assess such products. In terms of strategic foundation that we have been building for the past few years, a couple of years ago, we published 3 strategic pieces of work, 3 road mapping exercises, looking at first the human and animal gut microbiome, and there the focus was on dietary chemicals, dietary xenobiotics, and how those can affect the gut microbiome, which can lead to adverse health outcomes. Also, we looked at the environmental microbiome, such as from soil, water, plants, and so on, with One Health connections and food safety implications. Last but not least, we also published a roadmap on how omics methods, which I think paramount in order to be able to study and assess those products, could be used for microbiome-informed risk assessment, metagenomics being an obvious one, but of course other omics as well. All those aiming to build capacity in this area, and all those had a common objective, which was to do a systematic gap analysis and identify future actions, coordinated future actions in order to close those gaps. Okay, so how have we responded since then? 3 streams of action. One is to build the science base, and stemming from the road mapping work and through discussions and other activities, we will be funding directly 2 projects, one to develop standardized methods on metagenomics and the other one on biomarkers, but I will give you a bit more detail on the next slide on that. And also we have been trying to embed, let's say, food safety aspects into relevant EU projects and making sure that the food safety perspectives are taken into account. The second stream is through coordination at the European level. So for that, we have, let's say, mobilized the member states through our advisory forum, and through our network of national food safety authorities, to build a European picture and understand who does what and when. So we try to coordinate and align on that. Of course, with INRAE, I don't need to say much. It was said before, very much actively collaborating on the microbiome topic. And importantly, I think with the EMA, and there, the idea and the aim is to understand the shared challenges at the interface between medicinal and food and feed products. The third stream is the international engagement through the WNP collaboration, and then on the bilateral level with some key players such as FAO, and also US FDA. Okay, a little bit on the two projects, also to advertise for possible interested parties. So one will be on developing standardized metagenomics methods that can be used. So risk assessment, regulatory science grades. So it's not enough for us to have a nice metagenomics method. It needs to be robust enough to be used for safety assessment conclusions. The second project is on biomarkers. That will be in two phases. And the first project, which was published last week, and please have a look. I can share the links as well. Is to do a systematic appraisal of possible promising biomarkers coupled with in vitro assays and see which ones would be the best, which would have the higher likelihood of being applied in the risk assessment framework in the short to medium term, let's say. That hopefully will be followed by a second project, which will be to experimentally validate the predictions. So the second project was published last week. The metagenomics will be published in 10 days or something, hopefully. I think we're in good track to publish that. So what all this enables, what all this work can help us to achieve. So on the first point, so we heard about all those microbiome-based solutions, but for them to become of use, they need to be and reach the market, they need to be assessed. They need to be assessed for their safety. So I think that's a requirement that we cannot get away from. And EFSA has started to build the scientific tools to assess such products. To do that, we need a stronger evidence base. Carlos mentioned before about gaps. We are really having and facing a number of challenges that we need to resolve. There are a number of gaps that we need to close. We have started with the two projects I mentioned before. We're hoping that the colloquium will provide some recommendations for more actionable regulatory conclusions, and of course, the coalition of cohorts. I think the reference baseline data will be super helpful. Thirdly, and lastly, it's the need for harmonized regulatory science capacity. So we really need to get together and come up with consistent approaches. Key messages, and I close here. So we see that solutions, One Health, my microbiome solutions, are in the pipeline. We see that our frameworks are not able to cope with them, and we really need to work in this together. We have made the start, but that's no one single actor's job. It really is a coordination and joining forces that is needed. Yeah, if we want that such solutions reach their potential, we really need to make sure the regulatory science is there so we can assess them and they can reach the market. Yeah, we are committed, but for sure this is a shared challenge, guys. Thanks a lot.
Yes, perfect. Thank you, Konstantinos. Kostas. Is there any question? Yeah?
Yeah.
Thank you, Konstantinos, for the presentation. My question is about risks. What's best or worst between fast-tracking process to put products on the market and potentially having some modification to do, or being thoroughly cautious, so very long to put product on the market, and then to have no solutions available for all these needs that we are thinking, knowing that microbial-based products are probably not very risky compared to some drugs or other kind of pesticides and chemicals. Thanks.
Yeah. It's a good question. So, yeah, let's say.
I'll what do you call escalate it and answer that one for you, because the answer is very easy. You should ask your politicians. It's not up for me to decide. The level of risk that society is willing to take is what you elect your politicians to decide. That said, from our scientific side, we also need to be practical. We're all scientists, so and this is something I tell to our expert panels, we can keep up digging and digging and digging and digging to find more and more and more. We will never be finished. So at some point, we also need to tell from the risk assessment, this is what we have. We think it's good enough for you to make an informed decision. Now, if there is a 3% uncertainty and you're not willing to take it, then we need to continue digging and digging, and you will not get those products if you're happy to take the uncertainty and giving it a shot. But this is where I think it's really important, and we have in Europe, thank God, and let's keep it, a system where we have an independent scientific advice. We put it on the table, then someone else will decide, is it worth to go for it or not? So, sorry to step in, Costas.
No, that's fine.
We get a lot of these ones, and sometimes, if you're not careful, you will go further than what we should. So ask your politicians, and I know they are very eager here in this country, for example, to have that discussion. I agree. Let's hope others agree.
And just to add, I mean, it's also a matter of balance, yes. Depending on the intended use, you may be more thorough or less thorough. And I think the word pragmatic here is key. We're willing to do it. I mean, we try to do excellent science, but at the same time, we understand that, one has to treat different solutions in a different way.
I'll bounce on that, actually.
Yeah.
Microbiomes are actually coming into the development of new drugs.
Yes.
And this is completely new to obviously drug development. And one aspect that you mentioned is that you are starting to discuss potential narrowing aspects of regulatory aspects with EMA.
Yeah.
And so can you tell just a bit more about that?
Yes, of course. I think they are, as you know very well, Joel, they are dealing with an application, the microbiome-based therapy. So we have started, we have had a couple of meetings to share common challenges with regards to what minimum standards, let's say, we should have with regards to methods. One other thing, the concrete thing is that I will be hopefully going this year in a staff exchange program between the EU agencies with two EMA, and try to understand what they're struggling with. They came first, yes. I mean, we will be having applications soon at our table, I think. They have it already, so the idea is that we kind of understand and align with each other for sure on at least on what methods and standards we should be looking at.
I'd say if you see the potential, World Microbiome Partnership could be a provider of experts. I think I'm too involved for myself, but there might be a potential.
Absolutely. I have already highlighted to them, actually. Joel, yes.
Thank you for your talk. I mean, maybe it's much more a question for Carlos. I mean, do you have a practical example of this interconnection between all the agencies? I mean, the previous talk, I mean, you mentioned that you have a specific interagency One Health task force. I mean, do you have, proper example of this connection regarding microbiome?
I mean, probably not. I mean, microbiome might not be the best yet example where we see this. I mean, the task force, we have established this task force. I mean, the 5 agencies decided we need to get this going. And it has, I mean, I won't go through the details of all it right now. There are different activities within the task force, create more capacity among all the staff, but also doing joint risk assessments, for example. I think we just done an output. It's not microbiome, but it's also not irrelevant for microbiome on an antifungic, on the azoles, as some of you may know. So we looked at the azoles from all the perspectives together, the food safety, the public health, the chemical aspect, the environmental aspect. Microbiome is a topic that we will have to find the elements of unity between. We don't always need to be the five. Might not be sometimes the first topic, for example, the ECDC may be thinking of, since they're very focused on infectious disease, as you know, but we don't need everyone all the time on board. So the concept of the task force is not every five have to always chip in, but is to create a way of thinking and working, which is not built into the mentality of the agency. We were built, I mean, we are 25 years old, more or less, all of us. EA is a bit younger. We were built to do just our stuff. We couldn't care less about the neighbor. And that mentality has changed. One Health has helped to change it. So now we try to do this. So we've done it for azole, we're doing it for infectious diseases. We haven't come yet to microbiome. We would have to convince more of our colleagues. I mean, the environmental agency is probably easily on board. Exactly. EMA has a problem in their hands. For the chemical agency, they may look into it in hazard characterization, but this it's a bit, perhaps also a bit.
At the moment, they are a bit
exotic
yes,
for them.
Yes.
So yeah, it's not yet the mainframe example. But as we get more dossiers on the table, this will become an increasing issue for us as we get pressure also from civil society. Where are the solutions, and why don't we have them in the market? Right?
Yeah. I think EMA EFSA connection stronger. EA will come if we think of environmental microbiomes for sure. So those three, and I think it's a tiered approach as well. We start to engage as things become wider.
Thank you very much.
Thank you.
For the sake of time, we have to move to the next presentation. It's my pleasure to welcome Daniel Polensny from EMBL, who is going to talk about connectivity between different microbiome habitats.
Yes, thank you. Thanks, and thanks for giving me the opportunity to present our research here today. My work will be much more research focused compared to the previous presentations. I'm going to talk about the planetary microbiome, its structure, its connectivity, and gene flow across ecosystems, which we're very happy to see featured in this month's issue of the Cell Journal. And so we know that ecosystems across the planet are connected, and we know that microbes play a very important role in ecosystems, and that the microbiomes across our planet are also connected. Just to what extent microbial species and individual genes are moved between them and are shared between them is not well understood and not well quantified, which is why we set out to study this systematically from a planetary perspective. And so we started with three main assumptions based on things we've seen in the data so far. The first being that most microbes are environmental specific, so which would mean that a microbe that is very prominent and thrives in the ocean is not likely to be a commensal in the human gut, for example, but that there is interaction between nearby ecosystems. So there is sharing of species, there's sharing of genes, which we know, for example, from agriculture, where antibiotic resistances are enriched in soil and manure, can spread to nearby ecosystem and even bounce back to humans. So this is possible. And finally, that microbes transfer genes not only to their offspring, but also to between each other, to microbes in the same environment. And that includes the spread of antimicrobial resistance. And so we looked at this and we thought, what if there are microbes that are highly adapted, that can thrive in many different environments, and that can transport genes, not only their own, but also other microbes' genes, into very distant ecosystems and basically facilitate the flow of genes across the planet. And I will show you the framework we developed for this to study both the structure of the planetary microbiome, but also the exchange of species of genes and of antimicrobial resistances. So for this global analysis, there are hundreds of thousands of public metagenomes available. However, many of them actually lack basic annotations, contextual metadata, such as what environment they were sampled from, even in some cases, or for humans, what age or what sex the human was. So this makes reuse a little bit difficult, but our group kind of always tries to promote the fair data and fair data sharing and data annotation, and to improve the quality of data and metadata and the accessibility of these data, which is why we, on one hand, try to raise the standards in consortia like NFTI for Microbiota, which is a German infrastructure consortium on microbiota, but also in curating these public data sets ourselves systematically and making them accessible through databases that are then shared publicly. And with our databases for this analysis, we were able to dive deep into over 100,000 high-resolution shotgun metagenomic samples across the globe, which we systematically processed, systematically annotated, had basic contextual information for, and we're able to contextualize here. So if we take our first premise that most microbes are environment specific, this implies that because of this specificity, if we take the microbial composition and we cluster it in a specific way, we should be able to reconstruct where the microbiome was sampled from. And this was indeed true, and we were able to effectively impute some of the missing metadata in the databases by our approach, or in other cases, improve the resolution of the contextual information. And this resolved 40 of these environmental habitat clusters, which were broadly grouped into these 4 categories. So host-associated microbiome, like animals and humans, aquatic, ocean and rivers, terrestrial environments, and anthropogenic interfaces, which were wastewater treatment plants, skin contact surfaces, and built environments. And so we then wanted to look at what's the structure of the planetary microbiome. How is it organized? How's the sharing of species and genes between them? And this is what you see here, where each dot is one of the 40 habitat clusters, again, with the 4 major categories: host-associated, aquatic, terrestrial, and anthropogenic, where you can see that the lines are the number of species shared between them, you can see that the more similar the ecosystems are, the more species are shared. But you can also see already the impact of humans and human activity on the structuring of the planetary microbiome, because host-associated aquatic and terrestrial ecosystems are mainly connected via anthropogenic interfaces. So we already have the impact of humans on the connectivity of microbiomes globally. And so we wondered, can some microbes thrive broadly on this map? And can they connect basically all the different habitats globally? And this adaptability is called generalism. So we distinguished specialists, which have very narrow range of niches, microbial niches, and generalists, which can thrive broadly. And we quantified this systematically and found quite a few generalists, and then connected this to information on gene flow, so how horizontal gene transfer, how does one microbe transfer genes to another species, and how does this cross borders of ecosystems. So when we map this onto, it's a bit blurry, unfortunately. If we map this onto our network here and look at the left side, then I think you have to just believe me. If we look at the bars here, that in very similar ecosystems, the contribution of specialist and generalist is somewhat equal, whereas at very distant ecosystems, so basically connections from the human host to the soils or to the oceans, the contribution of generalists to this gene sharing is much greater than of specialists. So generalists can shuttle genes across very different ecosystems. And we showcase this using examples of AMR, where we have a multi-drug resistance island of 10 genes that we detected and saw in the generalist Aeromonas caviae, which is a well-known generalist and spread the gene to multiple Enterobacteriaceae. And we detected this island in the human gut, in wastewater treatment plants, but also in Lake Victoria near Kampala, Uganda, which at the time of sampling, it was clear that the sewage was discharged directly into the lake, so this was less surprising. But also at an estuary near Guangdong, China, just downstream a few kilometers of multiple wastewater treatment plants, which might be explained by the difficulty to actually treat Aeromonas in these plants, which is why probably this island persisted. So we know that there are global reservoirs and ecosystems on antimicrobial resistance. We know that there are vehicles for transmission that can kind of facilitate this spread. And so I think this is an opportunity for some monitoring or some further investigations, where, for example, from the WHO, we know that there's a list of priority microbes for AMR-related deaths. And maybe it's an opportunity to also look at these predictable interfaces of spread of genes, and to have a look at the three major bacterial families that we found that contribute to more than 60% of this long-distance horizontal gene transfer across ecosystems. And so here I've shown you the flow, the structure of the planetary microbiome, the flow of species and genes, but what we're also interested in currently and looking forward is how individual microbial strains and their genes move geographically at a more local scale. So we're looking at this through the TRACK expedition, which was led by EMBL, by our group, and many other institutions across Europe for the past 2 years that sampled soil, water, and sediment samples across the European coastline. And what you're seeing here is the strain connectivity, so basically microbial strains, so microbes that differ by only very few SNPs, so they likely have a biological, the same origin. And on the left, you can see I'm adding basically data on a country-by-country basis. You can see the nodes being the sampling sites, and the lines being the number of shared strains between these samples. And you can see that there's these hubs of very high connectivity of microbial strain sharing, which are disconnected, for example, in the Baltic compared to the Mediterranean, but show the connectivity and the spread of microbes on a local scale, but also on a European scale, and across soil, water, and sediment. And so we're very curious because we have this rich data set to see and look at metagenomics, metatranscriptomics, metabolomics. We're also integrating satellite data into this to get an understanding of the land use and the land use change over time, to have a full picture of the conditions in Europe and how genes can spread, what boundaries there are for the spread, and how we might tackle that. And here I'm showing you one of these satellite images, not in Europe, but basically a Sentinel-5P product where we see a sand or dust cloud raising on the Sahara across the Atlantic and splashing onto the Caribbean, which I show you just to give you an intuition of how the planetary microbiome is connected, one of the many, many ways it is connected, and how we should look at the microbiomes globally, because they are one microbiome, so to say. So with this, I'd like to thank you for your attention. First of all, thank Pierre Borg, my PI and dear mentor, who sadly passed away this January, my co-authors Chanyoung Kim and Jonas Schiller, and our entire group and funders. And thank you for your attention.
Thank you very much. Very interesting talk. Question?
Thanks a lot, Daniel. What about space? No, mankind is getting back to the moon, and we are talking to getting to Mars soon. Maybe. Maybe Elon Musk will be the first to travel there. Finger crossed. No, but any thoughts, any prospective ideas, any experience on microbiome in spatial environments, ISS, moon, meteorites, whatever?
No strong opinion on that. I think we still have fairly limited amount of data, and there's still methods being developed to actually monitor and see how the spread there is. So, yeah.
There is one interesting aspect that I cannot resist to mention. If you want to go to Mars, it will take 6 months to go. You can stay as much as you want, and then 6 months to go back. And 6 months for a space travel, it means that you need to circularize everything. You need to reutilize as much as you can of what you pull out, and you need to grow food. And so this question is really fantastic for the microbiologists, because microbes are everywhere in this context, actually, of circularization.
Great answer.
Not that I mean that we need to go to Mars.
Yeah, and I think we know that in those space station and so on, it's really a microbiome mess. One question and then.
Thanks for your presentation. So I have just one question. What about other species in microbiota? For example, fungi, virus, phage, are they shared also as bacteria?
Whether they are shared between with the ecosystem?
Virus, phage, fungi are shared as bacteria?
This is something we are currently looking into, and the answer is yes. I have no data to show you today on the work, but it's ongoing also in our group. We're looking beyond just bacteria into eukaryotes and basically surveillance and monitoring methods that could be used for different kingdoms. So yeah, definitely there's also, they're not silos, they're interacting, and we already have lots of associations of eukaryotes, of prokaryotes, of the different living organisms.
Thank you very much, Danielle. I mean, perhaps for me, it's more of a comment or a hope. I mean, I find especially interesting the dynamics, of course, and how you see mobility through the systems. I mean, in this case, the aquatic systems, but I guess the picture would have been painted the same on land as well, if that was the focus. And then, of course, now I'm putting again my regulatory hat. And then what I'm missing here, and I hope that scientists can help us to go, that is how do we link that to causal links into, you showed the antimicrobial resistance, how is this circulation of this microbes impacting resistant traits, for example, if I want to look into potential of microbiome being both a founder or a solution. And what I'm thinking about food is how this circulation impact security of food products. But even just on the environment, the capacity to replicate in a format that we can validate and harmonize, because it's a beautiful publication, it gives a lot of data, and then we need to find endpoints and pathways, I think Kostas has mentioned, for environmental microbiome degradation, and we're not there yet. So I just hope that HMBL can keep on and take us a little bit further to help us out. But thank you very much. Very interesting data, not to mention a bit scary.
Thank you. I have a short comment. So this is what I was trying to kind of allude to in my last slides, that now we're looking at the European scale with a very rich data set, so we don't rely on the public data with partial metadata and so on, but we have this huge cohort. And we hope that we'll be able to quantify, if we have a resistance gene, for example, in one environment, how far can it spread, at what speed can it spread, so we can assess risks better at the different interfaces.
Thank you very much for this presentation and answer to questions. We're now moving, and I would like you to welcome our next speaker, Thulani Makalanian, is that right? With professor at Stellenbosch University and founder of the African Microbiome Network. Thulani, thank you for your presentation.
Thanks so much, Emmanuelle. And I want to first start maybe by thanking you and Joelle for the invitation to speak. I'm hoping that through this talk, what I will make clear is essentially the importance of looking at African microbiomes. My talk will be a mixture of a little bit of advocacy, and then at the end, I will share one example, because I can't resist sharing some of the science that we work on, where we've done a project in collaboration with partners through funding that was enabled through the Horizon Framework project. So I am from the African Microbiome Group at Stellenbosch University, as Emmanuelle said, and our work is really using the One Health framework to underpin essentially all the research that we do. We've got a number of projects focused on human health, asking questions related to whether the diet that we eat is contributing to growth stunting and trying to find potential mechanisms of alleviating growth stunting. We've got projects that focus on animal health, trying to determine, for example, how livestock antimicrobial resistance it may be a reservoir for the spread of AMR in local communities. And we've got projects that focus on environmental health. One of the questions that we're increasingly asking is trying to understand how air pollution impacts on the long-term health of people based in industrial hubs along various African cities, and what the long-term implication of this will be. As you can see, for all of these projects, they focus on understanding the microbiology, and it is very clear increasingly that microbes underpin the One Health concept. If that point has not been made clear over the past couple of days, it should be increasingly clear, hopefully, through this talk. So one of the challenges of speaking after a number of people have spoken is that I will make points that have already been made, but I'll try and do this somewhat elegantly by skipping through some slides. But one thing that I will say here is that we know, of course, that there are various complications that make it difficult for us to get a true sense of microbial community diversity and functional attributes. There's been a talk that talked about, for example, horizontal gene acquisition, and all of these factors and dynamics make it very difficult for all of us to get a true sense of the relationship between microbial diversity and ecosystem function. What we know is that environmental conditions also disproportionately shape the phylogeny of microbial communities. And with this, I think we cannot really get a true sense of the role of the microbiota across various ecosystems if a number of systems, particularly those in Africa, are left understudied. So as I said, there's been lots of examples provided on the importance of how microbes underpin One Health, their relevance to climate change. We've recently written an article that talks about various solutions that can be deployed against the climate catastrophe. We've also, seen a lot of evidence talking about the role of microbes and infectious diseases. We've seen a lot of evidence more recently trying to show strong evidence of the fact that gut microbiota contribute to mental health, and there is an increasing evidence showing some of the mechanisms by which microbes do this. We know, of course, and there's been lots of discussions here about the role of microbes in food production and the importance in augmenting limited agricultural systems. And I've talked a little bit about the role of microbes as they relate to pollution. We know that increasingly the use of artificial intelligence is coming into play. We know that innovations such as AlphaFold are leading increasingly to increased use of microbial data in trying to understand more precisely mechanisms and finding new products that could be applied for biotechnological purposes. But the point that I would make is that in order to harness the microbiome for both public and ecosystem health, we need to build diverse training data sets that minimize bias and also prevent misleading or inaccurate system behavior. And we cannot do this, right, if the majority of existing data sets are left unstudied. So we know that we lack representative data sets. This is an image from a paper that we published a couple of years ago. What you can very clearly see is the lack of data on the African continent. We know that the majority of current available sequences focus on host-associated microbiomes. And what we've advocated for is for the establishment of the African Microbiome Project. Through this project, we are hoping that we can sequence more than 10 million samples in the next 10 years. It's a pretty ambitious target, and even more ambitious considering that we don't have funding to do this, but it will be done. So, and part of this is that we're advocating for focusing on clinical and environmental samples, and that some part of our rationale is that these data sets are quite important as we try and more implement the use of artificial intelligence systems. We're hoping to build the microbiome future that we all want to see and use. And in advocating for the use or the expansion of research on African microbiomes, one thing that we've called for is we've identified some of the core elements that will make it more feasible to expand research on African microbiomes. We've called, for example, for the establishment of coordinated research network, and we've got such networks that meet regularly on the African continent that are focused on trying to create capacity within the system. We've also called for the need to establish policy interventions. It's very nice being here because one thing that I increasingly learned in the past couple of days is that there are specific policies directing various aspects of the use of microbiome data, but in lots of countries on my continent is that we don't even necessarily have the framework to enable how research ought to be done across a variety of systems. And what we are calling for and working on through this network is we are trying to more and more try and write some of the policy documents that are required and promulgate these through the African Union so that they can act as an enabler for doing research on the African continent. We're calling, of course, for the establishment of a specific infrastructure development. And we're calling, in a sense, towards establishing what we are calling dedicated microbiome hubs that will be spread throughout the continent. And these will be dedicated platforms which will enable, for example, genomic sequencing that will lead to the expansion of African microbiome data sets across the continent. And as we do this, of course, we are calling for partnerships, partnerships through organizations such as the World Microbiome Partnership, and we are working with our partners in order to essentially think about how these partnerships should go forward, right? And we're doing this in a sense that we're not necessarily saying that we bring nothing to the table. I think there is a lot of a lot that African researchers bring, in a sense, not only in terms of enabling capacity to do research on the continent. We bring a lot of skills, some of them gained through people that have been trained in the best labs across the continent, but have gone back to the continent and are more intentional in ensuring that we start to generate not only microbiome data, but that these data are useful in order to develop the continent. And part of this is making sure that as we have these partnerships, we are working together with people in the EU and other parts of the world to enable these collaborative partnerships. So as I've said, what we would like is to have partnerships that will work towards expanding knowledge on African microbiomes. And that these partnerships are really crucial in order to enhance global insights on both public and ecosystem health. The point that I made earlier is that you cannot get to a point where you have a mechanistic understanding of microbial community function, whereas you do not know the vast majority of microbial communities across various understudied habitats, such as those that exist on the African continent. So, of course, these partnerships, as I said, mean that Africa should not just be a data source, but increasingly we are working on how we can become a knowledge generator. And I've highlighted, of course, the need in order to establish infrastructure, capacity development, and also what we would like to do is establish cross-disciplinary centers of excellence. And these cross-disciplinary centers of excellence are essentially the future of microbiome science. And these need to be co-developed with partners and not necessarily extracted from the African continent. So as I've said in the last few minutes that I have, I want to highlight one example where we've essentially applied the One Health framework in the microbiome work that we do. This is a project, and I'm showing this because, first of all, it's a beautiful image, but this image shows the research vessels, the Tawa Ocean, and it is a picture taken while the Tawa Ocean, the Tawa vessel was just off the coast of Cape Town in a project where we were interested in understanding how water inputs from large river basins of the African continent impact the biogeography or biogeochemistry of the South Atlantic. So for this project, We started off in Cape Town and essentially stopped at several points along the coast of Africa, including in Namibia, Angola, and I disembarked in the Congo-Brazzaville. And as part of this initiative, we collected samples that were used to not only establish the taxonomy of microbial communities, but these samples are being used now to gain, for example, insights on the extent of microplastic contamination off the west coast of the African continent. Understanding that we have one ocean. What happens in the South Atlantic, of course, has impacts on what happens in the North Atlantic. And for many years, these systems were essentially unstudied. So, of course, the research that we do and the system that we do most work on is the Southern Ocean, right? The Southern Ocean is probably the most consequential and important current system on the planet. It's important because it takes up over 40% of anthropogenic carbon dioxide. It also takes up about 75% of the total heat uptake. What we're trying to understand, we know that there's been a lot of work that's focused on the charismatic macrofauna. We've established a series of research projects trying to understand precisely the role of microbial communities in the Southern Ocean, understanding how they contribute towards carbon sequestration, biogeochemical recycling, and also in the sustenance of local and global food webs. So this shows essentially a research review that we wrote, and this underlies exactly the point that I made. The top bar shows the research that's focused on macrofauna, and the bottom shows the research that's done on microbiomes. What you can see is that when you compare the fact that I make the argument that microbiomes are so consequential, it actually does not make sense that we know so very little regarding the role of microbial communities and their impacts on these crucial ecosystems. So in this study that I highlight here, we essentially went on a cruise where we tried to understand the sea ice and sea water interchange, trying to investigate not only the taxonomy of microbial communities in these systems, but also trying to look at how they contribute to important biogeochemical cycles. In this case, we looked at genes that are implicated in the recycling of sulfur. And what you can see, without going into too much detail, is that when you look at this top bar, you can see that when you compare the levels of this toxic compound that's stored in sea ice, it's almost 30-fold higher compared to the levels of that exist in seawater. And what this research shows is that when you actually look at what happens in the South Atlantic and the DMSP levels that are stored in sea ice, they are potentially an important reservoir of this compound, and they could have important impacts in terms of acting as an additional feedback that may exacerbate the effects of climate change. And the taxonomic figure at the bottom actually shows that when you look at the genes that are implicated in the sequestration of this compound, it's actually harbored by several novel lineages that seem to drive the most extent of the cycling of this compound. So just in summary, what I've tried to highlight is that the work that we show, that I highlighted here, not only shows the importance of microbiomes and how they regulate the climate, but it outlines an important example of how when we work in partnerships with our collaborators in Europe, we can start to solve globally relevant and important questions using systems that are accessible from our side. That the second point that I wanted to make is that Africa is critical in order to also find global solutions, whether it's in the realm of food security, whether it's in the realm of climate change, or in understanding, for example, the growth and long-term growth of individuals in terms of health. We cannot adequately answer these questions if we are not understanding the core basis of the taxonomy and function of African microbiomes. We are working towards a point where we are starting to ask mechanistic and multi-omic type questions that are really important in terms of essentially laying a pathway forward in terms of the research that we all work on. With that, I'll bring my presentation to a close. I'm not sure how I did with time, but I'm happy to take questions if there are any. Thank you so much for listening.
Thank you so much to Lani for this great presentation and for bringing African issues in this meeting. For the sake of time, we will go to the next speaker, but we will have the roundtable so that people who have questions, just keep them in mind, and you will have time to ask to Lani. And so now we are welcoming Joël Doré, who is the acting president of the World Microbiome Partnership and a colleague from INRAE and a quite well-recognized scientist in the field of microbiome. Joël.
Good morning, everyone. I have it. Thank you. Good morning, everyone. It's a pleasure to be with you, and I'm really impressed, actually. I always feel frustration that the microbiomes are not as high as they should be in our thinking and in our minds and in the research proposals that are made, and I see a lot of it coming up this morning, so it's really pleasant. I'm here to pass the word on behalf of the World Microbiome Partnership, which we launched last year, as was mentioned by Philippe, Maugin earlier on. You've seen these slides. Humans are microbial in a microbial world, and I don't think I will center much on humans, although it's been my topic of research for a long time. WMP claims that microbiomes could be leveraged to help tackle planetary health issues globally, and the illustration just shows how much microbiomes are in the interaction between animals, plants, humans, and soil and agrosystems and nature at large. We humans are microbial, we are ecosystems, we are symbiosis. We interact on a constant basis, we adults, with 50 trillion bacteria, facing our 50 trillion human cells of the body. They're on the skin and at every mucosal interface of the human body. And in front of our 23,000 human genes, we have, on average, 600,000 microbial genes, which tells a lot of the potential functionalities that they will contribute, and a lot of the functionalities are actually essentially protective. What we have realized, though, is that at the time of the 6th global extinction, when we see insects, we see fishes, we see birds and mammals disappearing at the species level, well, there is an unseen loss of diversity in the gut microbiomes. And in humans, when we compare westernized population with people that have remained away from modernity up until today, it's a 50% loss of microbiome richness in Western populations. And that's with studies that have compared throughout the globe. And at the same time, what has been observed in terms of epidemiology is the rise of incidence, uncontrolled rise of incidence in chronic conditions. It's an epidemic. WHO really anticipated the fact that 1 human in 4 would be concerned by at least one of those conditions, and often several at the same time. And the curve is rising, sometimes exponentially. I just read recently that in the US, 1 newborn in 28 is at birth or will be in his life concerned by autism. This is really concerning, and we have to realize that taking into account microbiomes may help to make progress. So the way I like to present that is in healthy host microbiota symbiosis, there is a rich, diverse microbiota facing a resilient mucosal barrier, the monolayer of human cells that separates the microbial compartment and the human compartment. There is immune homeostasis and redox balance and lots of interactions. But upon genetic predisposition sometimes, but anyways, upon chronic perturbations with diet, with drugs, with pollution, then the system, the complex system, is fragile and may actually see alterations. We talk of a critical transition. We talk of tipping points that shift the system to an alternative stable state, essentially maintained by vicious circle of toxic interactions. So it actually challenges the change of paradigm that was mentioned at the very beginning for One Health, because this goes far beyond. If there is alternative stable states, then there is no continuum between health and disease. So you bounce in disease and the return may be very complicated. And you can apply that to climate change, to situations at the global level that really function the same. It's tipping points, and once you bounce, then the return will be very complicated. So it applies to human and animal health and plant health as well. And if you go there, then it's impaired functionalities, it's higher risk of chronic disease, and also, in some cases, resistance or non-response to therapy. So I just want to highlight the connection, humans in the center, soil at the right, and there are many, many similarities that we can point out. For changes and transitions, it's westernization of diet and lifestyle for humans, it's land use change and agricultural intensification for soil. And we have loss of richness in both cases. Mainly loss of bacteria for humans, mainly loss of fungi for the soil, loss of keystone taxa, loss of worm microbe interactions that are key to the system. Its impact at the functional level, loss of fiber breakdown ability, butyrate production ability. For the humans, it's loss of functions that relate to nitrogen or phosphorus cycling for soil or decomposition. And in both cases, loss of competitive exclusion ability, which means higher risk of infection for humans, but also for plants, for example, for the agrosystems. And in terms of socioeconomic impact, it's the rise of chronic disease that I mentioned, but for soil, it's a massive amount of impacts. It's loss of fertility, loss of food quality, loss of carbon fixation ability, and water retention, just as a few examples. So I think I have to be quick on that, bear with me. But in humans, what we have been able to describe is this situation where we have humans with low richness microbiota. It's 25% of the orange curve for obese people here. It's 10% in non-obese individuals. And we have individuals with high richness. And low richness is predictive. Predictive, which means a lot for clinicians, for example. It's predictive of non-response to calorie restriction in obesity, to reduce progression-free survival after immunotherapy in cancer. But it can be corrected by fecal microbiota transfer as an example of what we could do with this information. Many instances tell us that indeed the analysis of the microbiota can allow to predict the evolution of the system. For soil, what has been recently published, all the soil data is very, very recent, is that a microbiome may serve as a reliable indicator to assess ecosystem functioning under various soil and environment configurations. If you look at the top left here, it's a cropland. Well, in cropland, soil explains ecosystem performance as much as the soil property. The microbiome is as informative. It's half-half, 14% of variance explained in both cases. So it's a really nice indicator. And if we look at the impact of pesticides, then what we can say is that pesticides do impact taxonomy, the biodiversity of the community, but also functional groups and gene-based ecosystem functions. And the contribution of pesticides explains variation in soil biodiversity, and they show consistency or consistently a higher impact in croplands than all other ecosystems. So when agriculture is here, when intensive agriculture is here, then the impact is greater. It's a taxonomic impact on the composition, but also 1.7-fold more impact on the variance for functional groups and 5.5-fold more impact on functional genes within the ecosystem. So we see the impact quite a lot. So what we aim to do with the World Microbiome Partnership is to leverage the power of microbiomes to tackle challenges that concern all the aspects of One Health, basically. I cannot explain all that, but we have pointed a really key aspects that we want to investigate where microbiomes could be at the center. WMP is just an international forum connecting academia, industry, governments, and civil society to harmonize microbiome-based research for innovation, for animals all the way to humans, plants, and planetary health. And what we have proposed for the One Health Summit was this Inter-Health World Microbiome Observatory. So we want to really scan the various levels of microbiomes to leverage microbiome science with the World Microbiome Alliances for humans, for animals, for plants, for soil, for oceans. And we have started with the World Human Microbiome Cohort Alliance, building the reference for that, aiming to deliver microbiome-based monitoring tools and indicators of ecosystem health towards mitigation strategies with science-based evidence of impact on selected key challenges. And I will leave it here. Thank you for your attention, and you have the connection with our WMP if you want.
Thank you, Joël. You can stay here. Maybe you can join for the roundtable, which is not so wrong. I think we are missing one chair. Can you? Yeah. So thank you very much all for your great presentation. It was really interesting, and we have those new concept of evidence of real connection between the different habitats, which makes the problem a little bit more complex than we thought maybe, but that's very interesting for scientists at least. I will start with one question to you, Danielle, which is, okay, so you show this strong connectivity across ecosystem. And what is still missing to really understand and to be able to engineer, limit, use this connectivity between different ecosystems?
Yeah, thanks. So I think this is a bit what we discussed earlier, that we need to really be able to quantify the spread of genes and quantify the rates at which they spread, and have a better understanding of what ecological boundaries there exist that slow down the spread or accelerate the spread, like what the environment looks like that is very permissive for microbial strains or for individual genes, or not permissive, so that we can build for Europe basically a map of let's say permissiveness of microbes and of genes, so we know where the risk is and at what interfaces of environments we have a risk. And I think for this we also need to work on our data software or data integration, because as I've mentioned, we have lots of missing public data that could be used, and we need a data pipeline that we can ingest more and more data to kind of scan this at a global level, if possible.
Yeah, I think it makes a link with what Toulani presented in his presentation, in his talk, about this all this unknown about microbiome and microbiome habitats and environment. And so Toulani, I would like to ask you, how do you feel about this connectivity of ecosystem with those big gaps, and how we can improve that? And if you think that, if you've seen in the recent year changing in the way of collaborating with African country, for example, South Africa, and if it's more collaborative or more, with more equity, please.
That's a pretty tough question. But maybe to answer the question of connectivity, I think, Daniel's results show quite elegantly how we all live in an interconnected world, right? It doesn't make any sense as a scientist to narrow in and focus on questions that are so narrow with the exclusion of the potential implication beyond the area that you study, right? I think this is it's a nice message for microbes in a sense, right? Because this teaches us that even when we as scientists collaborate, our questions that we ask really depend on us having some synergy between us in order to more appropriately address those concerns. I'll give you one example. We talk about, for example, the role of the microbiome and its impact on food and growth of populations. But often a lot of those questions are inferred based on maybe not as clear insights by some of the scientists asking them. So I think when we move to a point where we are collaborating, we can actually enrich the type of questions that we are asking, ask more informative questions. That means that we are better serving the policymakers that fund our research because we are asking questions that have got global significance, but also ensuring that all of the stakeholders that ought to be part of answering those questions are on board. So I think there has been some shifts in the past few years, but we can all do better. And I think having conversations like these, thinking about exactly how we ought to collaborate, is really the good path forward in terms of what we can do. I hope I have addressed your question.
Yeah, thank you very much. Yeah, Costas.
Yeah.
So
Shift
you
of
are representing
attention.
the regulatory
Yeah.
aspects and
Yeah.
perspective. So for you, what are the main challenges in integrating microbiome-based evidence in risk assessment framework, and especially taking into account the complexity of those systems?
Yes, I mean, it's not easy to deal with complexity, even lower complexity when it comes to regulatory science. Why you want to be predictive, you want to be robust enough, you want to give a different answer. We had before a question: how much risk is acceptable risk? If we had the choice, we would say zero. Yes, but does it exist? No. So for products, let's say that kind of complex microbial mixtures, I mean, our system is based, as I was kind of, as I alluded to before, we have the chemicals and biologicals, let's say, type of products, yeah? So, but the whole thing, I should say, on the biologicals or the microbiologicals, were based on single strain. We want to know how much of it you are exposed, what does it mean, what the dose-response relationship, structure, yes? Microbiomes, they escape this structure. So that itself shows us that we have a fundamental shift that we need to make in the way that we do that, where we do risk assessment. So, and as I think I mentioned before in my talk, we need to move away from associations to understand how things happen, yes, and why they happen. So I think the mechanistic link, the causal link, is key for us. I think it's key for the community in general. Yeah. For sure, to have a regulatory answer, we must really know what adverse change means and how we can frame this. So those two things I would say would be there.
Okay, thank you. So just behind you, there is Pierre Mathieu on the screen. So Pierre Mathieu, you mentioned, thank you. You mentioned efforts or initiative to align with different organizations and to change the way of considering some of the questions. After this meeting and the scientific part, how do you feel about that, and what are your next priority in this kind of area concerning microbiome?
Yes, indeed. I mentioned the One Health task force that we built to bridge the different agency, chemical, medical, and so forth. I think it's indeed, as illustrated today, it's really the right move. I cannot predict the future, but I'm wondering if more integration should be done in further including more agencies into this task force, because those are the regulatory agencies, mostly the risk assessment one. But the one that makes the policy and drives the funding are not necessarily in this task force. They listen to their scientific assessment that they co-produce, but they should also be sitting at the table, I think, because they have different perspective, and having a broader reach for merging those different agencies could be of interest for Europe. So whether it's indeed the Commission, but probably the Parliament or other initiative like the EIC or other branch that we have that focus on microbiome support. And it was indeed very well highlighted during these discussions.
Thank you, Pierre. I have to say that it was a little bit. It can seem surprising to have on one side the European Commission having 100 million euro to support innovation in the field of microbiome, and on the other hand, EFSA saying that they don't know how to assess the solution and the product. So I think there is, I mean, we are all here to consider those gaps and to see how we can overcome them. I know innovation is taking 7 to 10 years, so we still have some time to do it, but we should tackle that. Thank you very much. Kostas, do you want to comment?
No, just say that I fully agree, and I think there is a very important gap there. Yes, we really need to kind of from the research, from the development, to making sure that if a product is going to make it into the market, it has to be safe. Yes, we talk about food product. Otherwise, it's just going to remain on the academic level with good data. I agree, all good, but not real use. So I think that gap, as you highlighted, Emmanuelle, is critical for me.
Okay, Joël. So you show in your presentation that microbiome diversity might be a marker for health, both for human and in the soil context. So can you comment on that? And how do you think what should be developed? What are the key priorities to make sure that those kind of information or results are embedded in future policies?
Yeah, so diversity has really come as a striking element. I cannot generalize fully. There are some ecosystems where diversity is not good. The human vagina, for example, in women is really not an ecosystem where you want diversity to support health. But in general, in natural systems, that's what we have seen. And probably it's just telling that that's what we've been able to document so far, but there's probably much more to discover. And actually, when we dig into finer details, obviously functionalities come next. So we first scan the microbiomes using composition assessments, and then diversity is really major. Probably it's an important support of the resilience of the system that can be stressed in various ways. I think that functionalities will come really very important as we move forward a better understanding of that. Now, the strategies we see coming up remain in the paradigm of a linearity between the composition and the risk or the presence of infectious agents, especially, and the risk of disease. And what I try to illustrate is that we have to open our chakra on that and be ready to address complexity much more than what we have been doing so far. And especially non-linearity, because I think that we have a lot of situations, and climate change actually was one telling us about that you bounce into an alternative state and it's stable, and the return may be very complicated, and it's true for humans, which I try to illustrate. Now, current initiatives are not leveraging the strategic importance of microbiomes. We are happy to discuss about microbiomes today, but the meeting we had, it was just a minimal aspect in terms of being addressed, but it's there underneath every of the aspects that were mentioned almost. So we have to yeah, to push. We are here to push the message. I'd say our priorities could be threefold. One, to educate on the importance of microbiomes as providers of protective functions, and that's what we do today, but we have to do more. Two, to promote the design of monitoring and surveillance tools. We see how better we could be able to do at various scales, actually. So this is upcoming, and we have to promote that, and we have to raise funds for that. And three, to deliver evidence of the benefits of leveraging science-based strategies to tackle selected issues. And for example, we link enriched soil microbiome functionalities in agroecological practices to higher yields and less chemical inputs. Well, we must demonstrate that this translates into stronger, more resilient host microbiome symbiosis in animals and humans. And so that's the link we have to be able to make. And I anticipate that there may be many local alimentary programs actually on the planet that could provide adequate context for this test of a hypothesis. And this is only one example.
Okay, thank you. Quite open. So I would like to know now, because I say that there will be some time for additional questions from the audience. Is there any question for those great guys? Yes?
Thank you. Thank you all for your presentations. I have a question, maybe, I don't know if it's a philosophical question. However, we have been spoken about One Health over the 2 last days, and today also linking with the microbiome. Well, there is a component, maybe it is considered implicitly, but we don't speak a lot about it, which is food. Because from animal to humans, or from soil to humans, there is something in the middle which also may impact the microbiome. So we don't speak a lot about it. I don't know why, and whether it is, however, covered by all the initiatives we heard about.
Yeah, I can address that, and I'll pass on the mic. But when we look at the health of the human microbiome, food is really a major element, and we have data, especially epidemiological data, that tell us protection and prevention will come with a Mediterranean diet, for example. And it's true for the Mediterranean regions, but it's true essentially worldwide if we apply the same types of composition of the diet that we ingest. And it's this continuum between diet, microbiome, and eventually health. And so that's one element I can bring to the debate, is that, yeah, we really see how food is central to human, but also to animal health, actually, in the system.
So maybe I can also tackle the question. I think you're right, maybe we don't speak about it enough, but I think there are lots of initiatives that look at the centrality of food and how that has both short and long-term health outcomes. Because classically, of course, in microbiology, we tend to focus on pathogens, that make you sick, but there's increasingly a large body of knowledge showing that what you eat has long-term impacts on human health, right? So we were chatting actually yesterday with Emmanuelle that they have a new initiative that's looking at childhood stunting and how you can augment existing foods in Africa and other places to ensure that these are fortified in order to ensure long-term circumvention of things like stunting, right? So there is a lot of research that's done in that field, but perhaps what we should do better is communicate what the research is and how we can better work with stakeholders in terms of supporting the current research and improving it.
I think was very well covered. Just to say that, Marianne, as Food Safety Authority, I did mention food, I think. I hope I did. But again, no, I see the point, and indeed, it's kind of, let's say, left a bit out. But as you know, we are also looking at dietary chemicals in food and how that can have effects on the microbiome, therefore the health of the host. And again, we see on the product side, we see the fermented foods being something that will be coming more and more. And again, the same issues, the same challenges apply there, but for sure not to be left out.
I could maybe also comment on the research side, because I think it's quite difficult to look at the connection, for example, of microbiomes, of gut microbiomes, and diet, because if you look at microbiomes, they are already quite personalized. So every individual has some strains that are unique for the person. And if we then consider diet, that's even more personalized. So you don't have the same diet day to day. So to get a good, high quality data set with which you can really find the connections of microbes and the microbial composition at a scale that you can make relevant comments on this association is fairly difficult, and therefore I think the data sets so far that have been produced are also not always openly shared, are limited in scale, and there certainly needs to be more efforts in this area.
Thanks a lot. I would add one thing, which is there is several startups which are considering to improve the soil microbiome and to restore soil by adding food for the microbiome, in fact, in the soil. So I think we can also have this in mind when we spoke about food or feeds, or it's a different concept. Now I would like all the audience to... you have one question? Okay, so last one. I like questions, so...
Thank you very much. I'm Miriam Aiteissa. I work in Action Against Hunger, an international NGO. We work on food and we work also on malnutrition, which is a health issue. I just... sorry. You can hear me better, maybe. I was just wondering whether if there were some links between all this science about microbiomes and antibiotic resistance that we are being speaking to. We have been sensitized to this for these 2 days.
I think I've tried to showcase some of the AMR-related effects that we see in the environment, that we see sharing of resistances. Okay, maybe you missed that part. That we see resistance sharing across different ecosystems and what the drivers are in this exchange. And so this is known, and we have published just recently something on this, but there's lots of research on this connection.
Okay, and now I would like you to join me to thank all those speakers from this morning. Very nice contribution and talks. And I invite Thierry Caquet, who is INRAE Vice President in charge of the international relationship, who will conclude this event.
Thank you, Emmanuelle. Good morning, dear colleagues. It's not an easy task to summarize and to conclude about all these experts. No. Yes, it works now. Thank you. Yeah, I was saying that it's not an even easy task to conclude or to summarize. I've picked up some ideas I will share with you, and we will have the opportunity to exchange afterwards during the lunch outside. First of all, I have the privilege to conclude this event. It was organized by the WMP, INRAE and EFSA, as you know. Thank you all in the room, but also those online. I don't know how many colleagues are connecting online, but there were also some people attending. That means that you're sharing interest in research perspective on microbiome, on the, let's say, practical use of this research, and also the regulatory issues. We discussed a little bit about this. And as you may have seen from this sample this morning, there's a great diversity of sectors, of geographical realities, but also a convergence of ambitions and objectives. And Personally, as the international vice president for INRAE, I'm deeply convinced of the necessity of gathering knowledge, gathering samples, gathering data to have the, let's say, the largest share of all this stuff to really obtain the transformation of many parts of our current life, maybe on health, on food production, and resilience or adaptation to climate change. And the Global Observatory for Microbiome Health that has been announced by Philippe Maugin in his introductory talk is a unique path toward global exchange, structuring microbiome studies on all continents. So if you have attention of what our speakers have said this morning, they showed how microbiomes, with some examples, Joel was saying it's only a few examples, one example, but we show that we can redesign our systems, food systems, but also health systems, to move towards greater sustainability and account for health at a global scale. We'll put it there, it's better. So, Eric Cardinale and Junxia with her presentation, stress out that enabling the knowledge of microbiome is a good way to move from reactive to proactive actions. It's very important. I'm come from the environmental domain. We are used to make some, let's say, forensics, say, oh, it is polluted, there's a lot of damage on biodiversity. We are showing effects, impacts, but do not always propose actions. So with all these approaches, we are now of the key, to some extent, to have some proactive actions and to be very, let's say, efficient as saving biodiversity, as increasing the sustainability of food systems. And Pierre Mathieu Pelissier and Konstantinos Paraskopoulos, sorry for pronouncing it, have shown that this challenge all the question of regulation. It was said that science progress very rapidly, but that regulation maybe lag behind to some extent. So we need really to think about how the European health policy, the European regulation can progress to ensure a common ground to provide, for example, new human microbiome-based therapies, push forward towards application of microbiome-based biocontrol products in agriculture or pollution remediation. The example of PFAS was mentioned by Pierre Mathieu Pelissier. Microbiomes also have the potential of becoming major indicators of public policy impact. We heard about the soil directive, for example, but it's not the unique one. For, let's say, chemical crop protection products, the monitoring of antimicrobial resistance, the question was raised, but there is a clear link between microbiome studies and IMR, and the soil health and pollution limitation. I come from the field of environmental science and community ecology, and I was very impressed by the presentation of Daniel regarding these insights about the exchanges, the fluxes between various types of ecosystems. Within ecosystems at a small scales, but also at a larger scale. And for me, it gives the opportunity to link the knowledge about the functioning, the structure, of biodiversity, what is there, are the exchanges, and the functional dimensions of biodiversity. As taxonomists, we are used to make inventories of species. Now with molecular tools, it's even easier for microbes. But who is doing what in the ecosystem, and what are the exchanges? And you gave us some insights of the progress we made, and for me, it's a new vision of planetary functioning. It's not only macroorganisms, but the microorganisms are very key in this perspective. And we need scientific cooperation, interdisciplinarity to demonstrate the impacts of microbiome research. And the World Partnership was mentioned, as I really appreciate the vision of Tulani Makalanian about the fact that the partnership is not a way to take some information, to grasp data and come back to our lab in the north. It's really a true partnership, should be knowledge generators, that the word you have been using. And for me, it's very important that is co-construction of this partnership with mutual benefits that will enhance the activity on these topics, as many other topics, of course, not only restricted to microbiome. The observatory we have been talking about will gather data, samples from all regions of the world, and to consider the specificities of ecosystems in these different regions, and not only to push forward global conclusion, but also to deliver recommendation locally adapted, dedicated to human, animals, and environment in specific regions. And we still need to demonstrate, I think, for the common public, the strong role of microbiome in food to demonstrate that this may be a source of innovation, of change of habits in food consumption, taking into account the food diversity in the different countries. It's not the idea is not to have uniform food, but to take the benefits of local food and the beneficial effects of local foods on microbiomes. I know that some work are currently ongoing, especially in South Africa on this topic, not only there, but in other part of Africa and elsewhere in the world. And probably that microbiome-friendly food will probably be one of the most effective weapons against disease, especially NCDs. It was talking a lot about NCDs yesterday and the day before at the Congress, because this is really an epidemic. It was also shown by some presentation. So we really need to have these allies with us, and food may be a strong allies. In some cases, it's not an ally, it's more an enemy of our health, but in some cases, we can use all the knowledge about microbiome and microbiome-friendly food to fight against these diseases. So the observatories should be a right tool to tackle all this subject, but also more. It's officially launched. There is a press release that was recently issued this morning about this. So now we have halfway has been done. We have collected so far under the control of Joël, 500,000 of microbiomes. So the target now is 1 million microbiome from man origin, for 2030. But it's also the way to enlarge the collection of microbiome, not restricted to humans, but starting rapidly to have all these microbiomes coming from the soils, coming from the marine ecosystems, from freshwater ecosystems, really an alliance on all microbiomes in this perspective. And having in mind that so far, some maps have been shown that around maybe 60 to 70% of human metagenome data are coming from North countries, and we really need to expand the collection of data and samples for the other part of the world, and especially taking into account Africa and South America, but also other parts of the Southern Hemisphere. The observatory is launched. It can soon become an international reference, like some genomic banks for cancer therapy. That's the idea behind. And understanding and making use of microbiomes needs a collective and structural engagement fully aligned with the One Health approach. So to conclude, I will thank you again all for sharing this ambitious goal, to share your knowledge, to share your work, and also to provide your support today, but also in the forthcoming years, because it's only a starting point, this observatory. We need to have everybody on board to share knowledge, to share data and samples, as I said. And we'll thank again our speakers today, both in the room and online, and also all the researchers of WMP that gathered their Human Microbiome Cohort Alliance, to all the participants, of course, of this meeting. And I will conclude by saying that all our gratitude to the OSH Fabric for hosting us today, and also the colleagues for À vous le direct for the technical support. Thank you again. And I will, of course, thank all the microbiome team from INRAE for organizing this meeting with EFSA. And I have a particular dedication to do for our colleague Indre. She's not there today, but she has a very good reason for this, because her daughter was born on Monday, on 6th of April. So it's a dedication for her and her baby. And for all of those of you that sometimes who have a lunch outside of the room, because I heard that we need to leave the room at 12:30 sharp. Thank you again for coming and for your attention.